Hein? Okay, so good morning, everyone. Apologies for the technical glitch, but let's start with a good note. So it is my pleasure to welcome you all to the MK1 auditorium on this significant occasion of World Zoonosis Day commemoration at the National Institute of Animal Biotechnology. World Zoonosis Day is celebrated every year on 6th of July to commemorate Louis Pasteur's work, the first injection of rabies vaccine. And that was happened in 1885. This day was established to raise awareness about zoonotic diseases. And you all know zoonotic diseases are those that jumps from animal to human. We are honored to have with us our director, Dr. Taru Sharma, whose leadership and vision continue to propel our institution forward. We are also delighted to extend a warm welcome to our distinguished guest speaker, Dr. Binoy K. Nandikuri, Director of CCMB. His expertise and insights are highly anticipated and will undoubtedly enrich our discussion today. About 60% of all human infections are estimated to have an animal origin. And all the recent and emerging infectious diseases of human are known to be almost 75% of those are known to be jumping from animal to human. And these statistics emphasizes that zoonotic diseases pose significant health risk and highlight the need for continued research and preventive measures. So together, let us make this day productive and inspiring as we share knowledge and commit to advancing the fight against zoonotic diseases. So now I would invite our director, Dr. Taru Sharma, to the podium to share her thoughts on this very day.
Thank you very much, Dr. Bapaditya Day. Greetings of the day. Good morning, one and all. Especially joining Bapaditya to welcome Dr. Vinay Nandikuri to NIAB for this talk. As he has already told, it was 6th July, and we requested Dr. Nandikuri. He immediately agreed. And then came I3C BRIC PhDs, and a bunch of us were away. And you were really kind and generous to shift the date. And that's how we are meeting today. Thank you very much for that. So as Dr. Bapaditya has set the stage for the zoonotic diseases and so on and so forth, I like to inform, especially to the director CCMB, besides many other things in the institute, we do have a bunch of laboratories who are focusing on some zoonotic importance of pathogens. And above all, we do have a One Health consortia, which is having 28 partners into it, cutting across veterinarians, medicos, and wildlife origin scientists. And some of the labs which are kind of really moving ahead with these zoonotic pathogens are Mycobacterium, your own area, Dr. Bapaditya's lab works, and of late he has shown through the genome sequence the Mycobacterium morages, and that work we have shown in our One Health consortia also. So the question there was whether it is a Mycobacterium bovis or Mycobacterium tuberculosis, but to our surprise, we understood it was morages. Well, not to stretch that story, then we have a group, another zoonotic importance bacteria, Girish works on brucellosis. And there is one scientist who works on a parasitic disease, which is also zoonotic in nature, that is Toxoplasma gondii. And another coordinator of this World Zoonosis Day celebration, Dr. Faisal's lab work on leptospirosis. So, I mean, without stretching more on what all we do on, maybe we can have separate discussion. But this is what is of importance within the institution which we do. It's not only the bacterial pathogens. We do have one or two laboratories, if not more, who are focusing on JV, which is again of a big concern, be it animal population or the porcine which is housed, or how it is being transmitted and dead end, it reaches to dead end, which is human. So with that, a couple of research leads which we have, and maybe we can find time when we interact with you over lunch or maybe over tea, and students of respective laboratories and other laboratories. So Dr. Vinay, we planned this day in such a way, the two coordinators, they work on zoonotic diseases, number one. And we have computer scientists whose data, as Bapaditya already told, we are all eagerly waiting to interact with you. Besides that, we just thought that if we could really get some of the posters displayed from those laboratories, so students will also feel belong to it, and this is how whatever it is. So I'll keep it short, and with that, I invite I don't know who's going to introduce officially. Yeah, please. Thank you so much.
So now I'll formally introduce Dr. Nandikudi. Dr. Nandikudi is currently serving as the director of Center for Cellular and Molecular Biology. Previously, and now as well, he served as a group leader and faculty at the National Institute of Immunology, New Delhi. And he is renowned for his contribution to the research on host pathogen interaction in tuberculosis. He is widely recognized for his pioneering research on kinase-mediated signaling networks in Mycobacterium tuberculosis. Dr. Nandikuri holds a master's degree in biotechnology from the Indian Institute of Technology, Bombay, and a PhD in biochemistry from Indian Institute of Science, Bangalore, and two successive postdoctoral fellowships at the University of Virginia and Texas A&M University, USA. In recognition of his substantial contribution to biosciences, Dr. Nandikuri has received numerous prestigious awards. He is a recipient of the Nashi Scopus Young Scientist Award in 2009, DBT National Bioscience Award for Career Development in 2010. He is an elected fellow of the National Academy of Sciences, India, and a member of the Guho Research Conference, among many. His current research focuses on understanding the complex molecular mechanism underlying infectious diseases, aiming to identify potential targets for drug development. Dr. Nandikuri's insights and discoveries continue to inspire and advance the field of molecular biology of tuberculosis. So I now invite Dr. Binay Nandikuri for sharing his invaluable research findings and opinion with us. Sir.
Thank you, Dr. Taru Sharma and Dr. Bapaditya, for kind words and the introduction and this chance to talk about our work and share our work with NIAB audience. We've been talking about that, visiting NIAB. I do come here once in a while for other official matters and even one or two dinners here, but hasn't realistically interacted or talked about our research with you people. In today's world, the new buzzword is obviously One Health. And that's because of the fact that most of the things, as we know, are moving from species to species. For example, even the COVID, SARS-CoV-2, has moved from species to ultimately reach humans, and many other diseases have jumped. So generally, the awareness with respect to research community in terms of how they view zoonotic diseases and human health has changed drastically, actually. Previously, those who were working on zoonotic diseases were literally a separate bunch from those who are working from human health related matters. That's not actually true anymore. You have rodents, you have pets, and you have farm animals, which actually can contribute to this transmission from animals to humans and vice versa. So the reality is the whole world, including water bodies, plant, and I mean environment basically involves many things, but certainly water bodies. And then you have this movement of things in an urban area where the distances between, things like pets and humans, or even otherwise animals and humans in the context of rural areas, in farms, pretty limited, given things are moving around. And so we have to have a better understanding of disease per se, infectious disease per se. What I am going to talk about has no zoonotic angle to it, but for me, it's the way we approach the problem is no different, whether it is a zoonotic disease or whether it is a human disease. So when we talk about something like tuberculosis, it's an old pathogen. It's nothing new about this particular pathogen. Generally speaking, since one tends to forget at the end, I start with acknowledgements, because here you see that, you don't do the work yourself. Most of the work that gets done and somebody like me gets to present happens because of the students who are really done all the bulk of the work and they're dedicated to what they do. So here is a bunch of students who already got PhD from my lab. And then you have postdoctoral researchers who have gone through my lab. And a couple of students from Yogender Singh's lab also worked in my lab. So it is a successful collaboration. I had given out 17 PhDs as of now and few other postdocs. And this is my current lab. I have around 7 students there and quite a lot of project fellows. And today I will be talking about some of the work by some of these students, not all. I'm not going to talk much about what we have done in the recent recent 5 years. I mean, maybe it's more like a story about how we deal with the problem. Tuberculosis is a pathogen that's something there for a while. It had many names, as you can see from this particular list. One, somewhere along the way, it is also called white plague, because people who get this infection tend to look pale. And at some point of time, it was kind of sought after because it was considered a fashion statement to look a bit pale. This was in the mid ages, not now. But point remains that tuberculosis has been there and continues to be an issue even today. People would not have known that tuberculosis is caused by Mycobacterium tuberculosis, except for this gentleman, Robert Koch. The thing about Robert Koch is that this is good enough, right? I can move around. Okay. The thing about Robert Koch is that he is actually a German bacteriologist who identified multiple diseases. Like he identified the reason for multiple diseases: anthrax, septicemia, tuberculosis, and cholera. He came up with this postulates which are even today as applicable. If something, an organism is causing the disease, you should be able to get the organism out. And if you get an organism out, you should be able to culture it separately outside. And if you use that culture and infect, there should be infection. And in such infection, again, you should be able to get the organism. If you can do this thing, it is the bacteria that you isolated, slash virus, whatever, you isolated in the first place, is actually responsible for the infection. This is the postulate that is golden and applicable to anything that we do. Mycobacterium tuberculosis per se is an ancient pathogen. As I was saying, you go back to Egyptian mummies, in their bones, some of them, you find Mycobacterium tuberculosis. And the bigger problem is the emergence of multi-drug resistant bacteria as the time progresses is the reason why it has become more problematic for us. MDR TB is not MDR and TB is a different matter. MDR, multi-drug resistance, is a problem when it comes to bacterial. This is not. I think then I will use this. Don't worry. So, but I think I am pretty audible no matter what. It will interfere. Too many mics. Okay. So Mycobacterium tuberculosis is there even in mummies, that you find that in their bones you'd see MTB infection. It's a old disease. Now we are talking about microbial resistance. Antimicrobial resistance is a problem. No matter whether it's TB, or you talk about simple gram-negative, gram-positive bacteria, wherever it is, multi-drug resistance is a problem. And it is compounded by the fact that in tuberculosis, the treatment is very long, 3 to 6 months for just TB infection. And if you're talking about MDR TB, the infections or the treatments last 9 months to 1 year or even longer at times. India has the highest burden of tuberculosis. Essentially, it's our problem to work. And we do have a highest infection rate and highest burden, right? We think tuberculosis infections are limited to lungs, but a lot of have this particular notion. You think of TB, then you would say, okay, most probably your lungs are getting infected. Reality is it's not lungs alone, but it is every other part of your body can also get infected. What is the percentage? Approximately 15% of the infections that are found, TB infections that are found in other bacteria. In fact, if you go and look into this, it can even affect the reproductive organs. And one of the sometimes when women have a problem in terms of conceiving, one of the things that ultimately given as even if you can't realistically identify the bacteria, is to give an treatment of anti-tuberculosis therapy for about 3 months, assuming that there may be a tuberculosis infection in the uterus. And it generally helps in quite a lot of cases. So these are the kind of things that tell you that it's not always limited to only the lungs. And in fact, other infections are quite problematic. The difference, like for example, you talk about Crohn's disease, which is also inflammatory bowel disease, inflammatory bowel syndrome. So you and the TB infection of the stomach have very similar symptoms, except that if you give an anti-TB, and then if you give a different kind of a treatment for Crohn's, because it's an inflammatory disease, you have to give anti-inflammatories. You give anti-inflammatory, and if it turns out to be TB, you're going to have greater level of TB, and you're going to aggravate the disease. So generally, when these things are not very clear, the diagnosis is not very, very clear. What they do is they give ATT for 3-4 months and or 2 months, and if it's not working, then they go for treating the Crohn's disease. Of course, now biomarkers are coming up, which would enable us to differentiate between Crohn's and TB, but it's still a challenge relative to the other. The transmission is quite simple. We know this because, in India, you generally have a lot of crowd in quite a lot of places. And over and above, you have a fantastic humidity level and temperature, which enables the transmission from a person who sneezes to the neighborhood people. And it is also a bit of an etiquette issue. Not everybody realistically closes their mouth properly enough. So that actually leaves aerosolic bacteria, which is alive for more than 24 hours. So it goes into the people, it goes into an unsuspecting host. There, it has fades. It has to can either get eliminated or become an infection. How does it happen? It is taken by the same cells, innate cells, which are supposed to eliminate. Tuberculosis thrives there. It somehow inhibits quite a lot of processes in the innate cells and tries to replicate there. But what happens? What happens under those circumstances is that it comes up with the next best thing. You surround it with other immune cells, okay, and thereby contain the infection. And this particular response of the host is basically what we call as granuloma formation. So these granulomas actually have 3 fates. One, you're fantastically very, very healthy person and you can really eliminate any pathogen coming near you. It becomes a calcified granuloma. There is no infection left. Else, it can become an active infection where your immunity is weak. It can become an active infection leading to full-blown infection, and eventually you become the transmitting person. The third is what we call as a latent infection, which happens in 95% of the cases, where the bacteria is not eliminated, nor the host allowing the disease to be established. What we call is a latency / I call it ceasefire. Right? They're all waiting for an opportunity. If you can eliminate it, you will eliminate it. If they can establish, they will establish. So when will they establish? When you become weaker. So somebody having an HIV infection makes it worse. Or suppose you have some elective surgery like knee replacement, or you have an arthritis, you go for an anti-arthritis, which is anti-inflammatories. All these things actually can bring up a latent infection. So These are some of the facts. Why do we continue to work on SARS-CoV-2? I'm sorry, tuberculosis. SARS-CoV-2, which is worldwide pandemic, which has killed almost 6.7 million official numbers. Call it as, even if you give and a take, it's around 10 million. TB every year kills 1.5 million. So, and it's been doing that for now ages. So given that, it is a pathogen that's not going that easily away. We have government mandate is to eliminate TB by 2025. At least you have to have tall strategies or tall kind of aim for us to see how far we can reach. So my own laboratory, I never worked on tuberculosis before I landed in India. I did my PhD in biochemistry and molecular biology, and I worked on DNA repair and protein translation. I went for postdoc. I worked on transcription factors in eukaryotes. At a later point of time, I worked on cancer. So when I wanted to come back to India, at that time I started looking for projects which are kind of relevant to what our problems are. And I kind of hung upon or stumbled upon TB as one of the possible things to work on. I was naive. I really didn't know how complicated this pathogen is, right? And I thought, and full of confidence, we can do it kind of a thing. So proposed a project to work on how ERK2 pathways in the host are modulated and what are the substrates that are of ERK2 kinase, MAP kinase are modulated upon infection. This was a straightforward project, but when I started the lab, I wanted to work on it. I realized the ground reality hit me that we do not have a BSL-3 facility and I can't do the experiment that I wanted to do. So how do you do it? When I started looking to the literature, I found that some kinases and Mycobacterium tuberculosis also have serine threonine kinase. I was a signaling man. So eukaryotes have 800 kinases, serine, threonine, and tyrosine kinases at the membrane or intracellular. Mycobacterium tuberculosis has 11 of them. And around that time, there was a paper that was published in Science, which talked about a kinase named PKNG being very important for the survival of the pathogen inside the host. So that basically made me, okay, let me try this out. Got to know that Yogendra Singh and Pradeep Chakravarti are already working on it. But I was young, I was like, fine, doesn't matter. Competition is no issue. So it nevertheless got into it. And Ibai Singh became my collaborator. And I generally don't worry about what the competition is. Just go ahead and do whatever you feel like doing. But collaboration is good. So serine, threonine, kinases and phosphatases is what I worked on for a long while. And when you're working on something like a kinase, you're going to have targets. You're going to have proteins which are downstream, right? And that is what led to my work on peptidoglycan synthesis, cysteine metabolism, and regulation of cell division, evolution of drug risk. This is a different project, but many of the other things. There are two projects which were very distinct from what we do. One of them is the evolution of drug resistant and host-directed therapy. Also, there is a nucleoporin phosphorylation. Nucleoporin phosphorylation was my carryover work from postdoc, which I stopped after 10 years. So one student worked on it, nobody else did. And this is where we were for all these years. I'll just talk briefly about what I'll do is I'll try to give a glimpse of our walkthrough. I mean, what kind of work that I have done, rather than say one story, which is what I generally do. But this is a longer talk. I thought I would do it differently. Cell signaling is a process, all of you know. Simple thing. You have a signal at the cell and you have a response inside. And this happens through either serine, threonine, or tyrosine phosphorylation in eukaryotes. In 1990, when they started, genome sequences started becoming available, they realized that there are serine, threonine kinases also in bacteria. And so otherwise, what does bacteria have? Bacteria have something called two-component signaling. What is two-component signaling? A histidine kinase, which is at the membrane, and a response regulator, which generally is a transcription factor. Histidine kinase, the name suggests, is histidine that gets phosphorylated. And the response regulator is aspartic acid, which gets phosphorylated, and the transfer is from histidine to aspartic acid. Let me tell you, both are labile. Means these bonds are not very stable compared to, like, they're covalent, but they're not very stable bonds. And there are also phosphatase activity involved here. This is what most of the pathogens or most of the bacteria use. For example, E. coli have 62 component signaling kinases. They can sense the environment and respond just using the 2-component signaling kinases, right? And 2-component signaling system, not kinases. On the other hand, serine kinases are essentially a very prevalent signaling system where you don't have one substrate. So it is almost a different scenario here. You think you are doing a 100-meter relay run, not 100-meter, 400-meter relay run. After every 100 meters, you are supposed to pass on the baton to the next person. That's how a relay happens, right? You've seen the relays in Olympics. Now think of it, the person running first one is giving 3 batons in the second, and now the person is giving in third level to 3 or 4 people. Means you are doing a system where you are expanding. That's the difference. Here, it's not one-to-one signaling. Here, the signaling expands at the, like, once you get on the surface, and it's a cascade. That is what serine kinases in eukaryotes do. How do they do it? They phosphorylate this other kinases, which phosphorylate other kinases, like that it goes on. ERK2, for example, is a cascade. It starts with RAF, MEK, ERK, RSK. It keeps on moving. So what is common about serine kinases is it has 2 particular lobes, the N-terminal lobe and the C-terminal lobe. It's a snowman without the head. Okay. And there is a connecting region called activation loop. So this activation loop is like a loop that holds these 2 lobes together. And when the phosphorylation happens, they're held properly. So the activity increases by approximately 100-fold. So a kinase is inactive, active. And the phosphorylation is on serine, threonine, or tyrosine, and they are not labile. They cannot be broken back unless there is another enzyme called a phosphatases comes into play. So it's a system of kinases and phosphatases that regulate the machinery in eukaryotes, right? So what happens in Mycobacterium? There is one phosphatase, PSTP, and there are 2 tyrosine phosphatases, PTP and PTPB, and 1 tyrosine kinase, plus 11 serine, threonine, and protein kinases. So one thing common about all these kinases, leaving one of them, they are at the membrane. And the kinase domain is inside, and the extracellular domain is outside. The moment extracellular domain is outside, it is taking in the signal. The kinase domain is responding. Only one kinase is different and exclusive on this. It's called PKNG, which is completely soluble kinase, which is somewhere here. Okay. My lab has been working specifically on PKN, PKN, and PKN. We have worked on other kinases, but one paper here, one paper there. Here, we have worked consistently. For like years, we have been following project after project on these things. So, protein kinase A and protein kinase B are the only kinases which are essential for the bacterial survival. What I mean by is, you take that out, bacteria is going to die, right? So it is in the same operon which has CSPP, which is the only essential serine threonine phosphatase. We have worked on all 3 of them. We have actually generated conditional replacement mutant of PSPP. We have generated a conditional replacement mutant of protein kinase A, and we have borrowed or rather collaborated with somebody else to take a conditional replacement mutant of PKNB. What is a conditional replacement mutant? You provide an inducible copy elsewhere, knock out the origin. So now your kinase is there normally, but when you add an inducer, either it shuts off or not there normally. When you add an inducer, it shuts off. It turns on. So this is what we use for all this kind of work. And around the time when we were working, there was another paper published by Robert Houston's group in this thing, which showed overexpressing either PKNA or PKNB leads to very aberrant cell shapes. So the thought process is that these kinases are involved in cell division and cell wall synthesis. Nobody really knew how. PKNB has a very long extracellular domain. And the thought process was this, that, before I do, how did we prove essentiality? we published 2 papers in JBC around that time. We showed that if you take out pKNA or pKNB, in both cases, you do not get any infection at all. So these were conditional mutants. So you give doxycycline in the water to the mice, right? Something like that. So that time we showed this, but then we started looking into what happens in case of pKNB, and there was a hypothesis that was again coming from the earlier work by Robert Hussain and Tom Albert, that there is a ligand is a peptidoglycan fragment called neuropeptide, and when it binds, the kinase would most probably become active and phosphorylate its kinase domain. Means it's inactive, binding of the ligand makes it active. This was the hypothesis. We tested this hypothesis by identifying the residues in collaboration with Debashish Mohanty, which interact with the ligand, and we made a mutant of pKNB. And what our work showed is something very different. Normally, protein kinase B is actually localized to the pole. And in Mycobacterium tuberculosis, the growth is from pole. Peptidoglycans are made at the pole. And what happens is the ligand is lipid 2. We proved that. And what you see is a regulated phosphorylation of pKNB and proper life. There is no problem. If I take away the ligand binding, what we showed is very contrary to the original hypothesis. We showed that in the absence of ligand binding, the kinase becomes hyperactive, not lower, hyperactive, and it loses its localization. Once it loses its localization and it becomes hyperactive, it phosphorylates substrates indiscriminately, eventually killing the pathogen. Means it's important to keep it regulated either at lower end or at a higher end. So in the absence of that, you end up getting the death. This is pretty much contrary to what we believe in terms of kinase field, ligand binding activation. We are saying no ligand binding, more activation, right? So we also were working on protein kinase G. At that time, it has a very 3 distinct domains: kinase domain, and a domain for interacting with the protein, and then an terminal domain called TRX domain. So the N-terminal domain also has 2 CXXC residues, which are involved in, generally these kind of residues are involved in redox sensitization. But what we basically did, around that time, there was also a paper, which I said the 2000 paper, the paper that got me into this. You take out pKNG, you don't get lysosomal phagosomal fusion. Pathogen survives in the phagosome. If it fuses with the lysosome, pathogen gets eliminated. So what we had actually, they have shown is that pKNG, in the absence of pKNG, more of these fusions happen, more of the elimination happens. So it is important for the survival. The question is how? What we showed with time is that when you do not have pKNG, the TCA cycle actually gets affected. Because there is a protein called GARE, which is regulating multiple enzymes in the TCA cycle. Phosphorylation of the GARE actually is a regulatory phenomena that allows the TCA cycle to be more effective. We actually did a lot of experiments and showed that protein kinase G is important for maintaining this redox balance and maintaining the TCA cycle level. So we went on to show this further, that it is actually involved in latency, because the metabolism is a very critical phenomena when the conditions are different, which is latency. Latency is the scenario where you are surviving in a granuloma. What we showed is if you do not have pKNG, latency establishment is not easy. In fact, we did this experiment in a mouse, a year-long experiment done by Mehak. I'll show her photograph in a while, which showed that protein kinase G is required for establishing latency in the bacteria. So what it basically means that if we can give a therapy which includes the existing drugs for something that is going to inhibit pKNG, you should be able to eliminate latency. Did I test it out? No. We have done it in mice and left it there. And that's because I'm more of an seriously mechanistic and tinkering guy, not the one who looks at therapy. But these papers are out. So this work is all done by, the one which I just now showed is done by this girl, Mehak. Very talented student. After finishing that work, she came to me and said, I'm trying to identify the transcription factors that are going to have an impact on pKNG, this and that. I said, okay, do it. Generally, I don't stop students from exploring things outside what occurs. There are no boundaries for exploration. So she said, I'll make it. She came back to me with some other thing and she said, this particular transcription factor is most probably going to control PKNG. And she started working on it. 3 years later, I asked her, what was the PKNG connection? She didn't remember. Okay, so eventually it became a transcription factor story. So that's how I actually landed in a transcription factor, because a student said there is a connection and there was no connection. So eventually it's fine. What we have in Mycobacterium tuberculosis is 200 transcription factors, approximately, out of which only 10 of them are essential in vivo, means outside you grow, nothing happens, you take it out. But you grow inside a mice or in a macrophage infection, they are essential. That's what high throughput data say. Out of which only one of them is unique to actinomycetes. So we chase this. Okay. So the transcription factor is a transcription called RV1332, part of 11 gene OPROM. Okay. And this is the second one. And it is a very small transcription factor, 218 amino acid, non-essential, essential in vivo, ex vivo essential, in vivo essential, and unique to actinomycetes. But what you notice is there are 3 genes called MEC, CISO, SysM. These are involved in cysteine biosynthesis. So what we did is we basically knocked out, or she knocked out RV1332 and created a complementation string. You have to have a complementation where you put it back. After doing that, she looked into the growth. There was no difference. There is a looked into the growth in macrophages, and you can see a very clear compromise whenever you knock out. And when you look into the mice, same thing. So it is very clearly proving what's known. Question is mechanism, right? So we went and looked into what is actually affecting. When you go into the host, multiple stresses are there. So we looked into each stress individually, and we showed that it is the oxidative and nitrosative stress that compromise the survival of the mutant. So if you have oxidative stress coming from multiple ways, whether it's CHP, H2O2, diamide, in all these cases, you see that there is a significant death of the pathogen. Question was how? Again, it's a fantastic story, very long one. I'm going to summarize it literally. But what we did is that we basically looked into without oxidative stress, what happens, and we found that in the absence of oxidative stress, which is interferon gamma null mild, you have a complete survival of the mutant is as good as the wild type. So it is very clear oxidative stress is where the issue is. Now the question is how, right? To do that, we actually had to do a lot of experiments, but one of them was obviously RT-PCR. And before that, we were looking into what I was saying, cysteine biosynthesis pathway. In bacteria, cysteine is produced through 3 different pathways. One of them is from the serine, which is canonical pathway, means not always exists. And there are 2 non-canonical pathways where it is produced from O-phospho-L-serine. One of them is by this particular pathway, MEC-CISO-SysM. You can see first there is SysM is a synthase that allows formation of cystocysteine. Cysto is a protein, mind you. It binds to a marks and adapt, and then it becomes hydrolyzed and become L-cysteine and this thing. Because it forms an adduct, it is a fantastic pathway under oxidative stress because it is stable. Otherwise, cysteine is never that stable. So it is required for multiple things, cysteine. What we showed is that normally speaking, these 3 genes are upregulated in oxidative stress. And we showed it by both RT-PCR and Western blot. We identified a novel interacting partner that's called a sigma factor, sigma H. I'll talk a bit about sigma factors just in a while. But what I'm trying to say is that sigma factors also, alternate sigma factors also respond to stress. Sigma H is known to respond to oxidative stress. So what we showed was that sigma H under oxidative stress interacts with the transcription factor. If I don't have sigma H or if I don't have transcription factor, in both cases phenotypes are same. Right? What we showed was eventually the bottom line is this. What we showed is that MECO, CISO, SysM, these are all transcribed, but at a certain level, normally. In oxidative stress, the transcription levels of these MECO system go up because it has an additional regulated transcription specifically meant for it. Okay, and that is important because it has to produce a lot of antioxidants. And this happens through a transcription factor called AOSR, which becomes a disulfide bond, interacts with a sigma H and interacts with RNA polymerase, goes and finds an alternate promoter. These things are all shown. Eventually, what we end up getting is how you control a cysteine metabolism. We have taken this story further by creating the system knockout and alternate cysteine synthesis knockouts, but I'm not going to talk about that today. But while I'm at Sigma Factors, what I do in the lab is that I generally spread out multiple projects and say, okay, whatever, when a student comes in, you can pick on this project. So this guy, Biplab, has done work on Sigma Factors. And what we have as a Sigma Factors, all of you know about literature, right? Sigma Factors are important for what? Promoter binding, promoter recognition. So they recognize -10, -35. What do? They bring in polymerase. Without that, nothing happens. So how do you recognize this? You see this here. There are domains that are there in Sigma Factors which recognize -35. There are domains that recognize -10. There are domains that recognize both sides, the additional thing. Okay. In Mycobacterium tuberculosis, there are 13 Sigma Factors, but only one housekeeping Sigma factor, what we call a Sigma A, and there is which has all domains that are necessary. What we showed, is that the Sigma A it's already this is a chiral structure was available in the literature. So we knew what and all interact where. What we had to do is that essential for the survival of pathogen, complex structure is available, no studies on in vivo. So we did the in vivo. We generated a conditional knockout of Sigma A. Conditional, again, the same thing, put a copy somewhere, knock out the original. Easier said than done. So it's basically very easy when you start talking about your generated knockout. Take it from me 6 months to 1 year straight away, because doesn't work one shot. Students struggle, they get frustrated, and then eventually things somewhere, you keep on saying that, no, it will work, try once again, kind of a thing. Eventually it works. After that, they think that their project moves. Most of the time, you can see when we don't have, and we add anhydrocycline, it's a complete shutdown of the Sigma A from the additional process. What we showed, we did an ex vivo experiment, infection experiments, and what we showed is that this is necessary both in an starting itself. If you don't have Sigma A, you die, there is a death, and you allow an infection to establish and deplete Sigma A, then also you have death. So Sigma A is important at every stage of bacterial growth. It's not a very complex one. It's a Sigma factor. It's expected, right? So we also looked into the transcriptome analysis, and we identified that there are something like 2000 genes, which is 50% of MTB is transcriptome changes when you take out the Sigma A. And we went on to show that it is very unique transcriptome changes, not overlapping with other Sigma factors, right? And we are then tried to show what is the domain that is important. And we showed that the N-terminal region, which is extra, is important for Mycobacterium tuberculosis Sigma A function, because this particular deletion mutant, up to 132 it functions, 130 to 179 you delete it, dies. So we showed many things, what kind of regulation happens and all. We are still carrying on this project in collaboration with B. Gopal from IISc. We are looking into liquid-liquid phase separation. Liquid-liquid resolution still having some kind of an complexes in the cell. In how in bacterium, we are looking into its interacting partner, novel interacting partner. We showed that it interacts with IF3, the translational machinery, and we are looking into how that is important in the context of Sigma A function. All these things are being looked at. So that's the scenario where we are in terms of Sigma A. So these are briefly the stories that were already published and done again by the as I said, talented graduate students. But I'm going to talk now briefly about some unpublished stories. 15 minutes, right? What I'm going to show you is something that we have not published yet, something that is ongoing, that is something that is exciting. So, cell division. A cell divides, one becomes two, right? Bacteria divides, or human cell divides, everybody divides. Now, when the cell division happens, if it's a bacteria, you have to constantly synthesize the cell wall, right? In E. coli, synthesis happens across, everywhere. In mycobacterium tuberculosis, it happens from the ends. Okay, and second thing is the speed at which the synthesis happens from the ends, not the same, right? So this is all work done by my graduate student, Yogita Kapoor, who's still in the lab. She's finished 5 years in collaboration with Thomas Pukadile and his student Himani, Siddesh and Arnab, again from ISER, Pune. And this is Debatri. I have not written a name, but basically that her name is Debatri. This is Tej and Nitesh. They're from CCMD. They have done the RNA sequencing analysis. A photograph that is missing, and a collaborator of mine is Neeraj. And Neeraj is actually in Canada. My student has gone to his lab and did the experiments and come back. And so fantastic data. Now we have to just send it back because we are getting kicked around a bit by the reviewers on this. So tuberculosis, this division is not equivalent. Now the growth is equivalent. What it leads to is a smaller cell and a larger cell, asymmetric cell division. What's the advantage? One can do is go towards what we call as persisters or refractory cells, right? How does it work? You have growth coming from both poles, except that this is one pole is a fast growth, which is generally the old pole. Newer pole is slower growth. And eventually what happens when the cell division happens, this becomes old pole. So the next time this one grows faster, this one will continue to grow faster, and you have always a bunch of smaller cells, and you have a larger cell. And that actually is important when you start subjecting the bacteria to antibiotics. The one which is growing slower eventually becomes refractory to antibiotics. And that is called persistent cells. They're not resistant, refractory. Means उनको कुछ फर्क नहीं पड़ता. They reach a point where they are refractory to the antibiotic conjugate. And when you actually take out the antibiotic, they back up. And they're not resistant to antibiotic. Next time you add antibiotic, again they go to such a state. They're called persistent. Eventually it can go to this thing. So there is a protein called VAC31. Murji, these are the things which are involved in mycolic acid glycan synthesis, which are all localized to the cells. And LAM is a cell wall protein, cell division protein. VAC31, one of the protein is essentially what we call as a scaffold. Scaffold, you know the word scaffold comes from, have you ever seen the construction of buildings that happen? You see outside long rods and other things which is used for painting, plastering. That's a scaffold. Without that scaffold, you can't do the. So scaffold is a support that brings two things together so that the work gets done. Got it? So that is what the function of the 4A protein called VAC31 is. So what it does is that it has a long coil domain in the C terminus and a small domain called D4A domain in the C terminus. What is a D4A domain? It is essentially a heptad. It's a heptad which is consisting of histidine acceptance, which is basically repeats are multiple times present, and these are required for the protein interactions and other things. So this is the structure. It's not a monomer, it's a multimer. Okay. So point is, how does it do it? People shown already that if you take out div4A, that is VAC31, you end up getting a football. You will end up getting a big cell which doesn't divide well, and eventually after around 4 cell cycles it dies. This is all shown, nothing new. Point was mechanism. How? Right? So what we started off with we generated our own conditional knockout in smegmatis, not tuberculosis. Life is easy. One is 21 days, the other one goes into 3 days. So this is how it looks when you actually take out VAC31. It looks like a ballooned up place, and eventually in about next 2 cycles it will rupture. That's because all your peptidoglycan synthesis, your mycolic acid synthesis, everything gets affected. Localizations change, everything really in bad shape. So she started looking at, surprisingly nobody has looked at TEM, transmission electron microscopy pictures of it. She started looking at it. I mean, completely her own initiative. Okay. With ATC, smegmatis looks like this, nothing, no difference. What you see inside is the white patches. Okay. Focus on that. This is our knockout loop. Okay. When we got this picture first time, we were really not sure what it meant. But our assumption is generally white dot lipid body. So obviously we did the next best thing. We looked into lipid bodies. So how do you look into it? There are fluorescent dye called body P. You add that, it goes and binds to wherever the lipids are. You can see how the lipids look in wild type, in the mutant with the VAC31 without. There is so much lipid that is accumulated. So now lipid accumulation is not unusual. Your own cells accumulate lipid. Bacterial cells accumulate lipid. Where do they accumulate? Anytime the cell is stressed, one of the things is store some lipid. It is a kind of a reaction to everything. But why and how? Right? So it is not only the depletion, but even a overexpression leads to lipid accumulation. So what does it tell you? You have a scaffold. You have too much scaffold, the person can't climb, right? It becomes too messy. Too little a scaffold, again you can't climb. So it's essentially a scenario, you need to have right level of scaffold, protein concentration inside the cell for it to give you a proper phenotype. Either deplete or overexpress. In both cases, this is overexpression. In both cases, you have an issue. We went ahead and did the interactome. We did a lot of other things. I don't want to talk about it. And we identified that many proteins that are involved in cell wall synthesis and lipid metabolism and many other processes. So they are my top 25. In 5 independent experiments, they are my top 25 hits. And then we looked into it. In mycobacterium cell, there are 2 kinds of membranes. Something basically called IMD, intermembrane domain, which is where most of the synthesis, these are all near the pole, but there are some domains, subdomains in the membrane. It was established. We find that quite a lot of IMD protein and cell wall proteins bind with the VAC31, right? Then we started asking this question, can they directly bind to lipid? This is where Thomas Pucadile comes in. He's the master at it. So I sent my student to his lab. What they have done is very interesting experiment. You have his-tagged VAC31 and they put a liposome. In the liposome, they put red fluorescence labeled liposome. In this, basically, you have this particular lipid, which when it is present, eventually cross-links, and you will know whether it is binding to the lipid or not. Right? So when you increase the covalent linking, eventually when you run it on an SDS page, you will still see if the fluorescent tag thing is bound or not. So this is the assay that was standardized by them long ago. They simply did it for us with 5 different lipids. PG, okay, and PG, PE, PG, and CL. CL is cardiolipin, okay. So all these things we do, and we find that it is only binding to cardiolipin, not to the other PE or PG lipids, right? So how does it bind? VAC31 has an N-terminal region which has a dipore domain, and it has a K lysine residue that was shown by others to be important. We simply made 2 mutants and looked into whether the binding changes. The deletion of the N-terminal region decreases the binding but doesn't abrogate. The deletion of a single point mutant doesn't make any difference. You may be wondering why the migration is different. Understand this, even in an SDS page, sometimes even a single amino acid mutant can change the migration. Why? Because though in SDS page it's supposed to be denatured, take it from me, it is not completely denatured. Unless you add urea to the gel. Okay, in which case it will be denatured. Understand this, right? We did the colocalization to show that the liposomes are actually binding with the GFP tag protein, they're colocalizing it. So these things are all confirming that the binding happens. But we were not convinced that binding is actually doing something. Thomas actually came up with another experiment. He said, maybe your protein is tethering. What is a tether? Tether, as the name suggests, is holding one place, holding the other end, bringing them together. That's a tether. Okay, I bind to the lipid, another one binds to the lipid. You're a dimer, bring them together. That's the tether. Okay, so he did this. He basically said maybe it's a tether. So basically, let's look at it. So what he did is an experiment where cardiolipin liposomes were basically looked at with a microscope normally, and here it is how it looks. Do you see anything? They're all same size fragments, correct? Now the second one, in the presence of VAC31, what happened? They're all much bigger like lipids. Why? They're coming together. So it is most likely a tether. So what happens with the mutant, right? The mutant, you can see very clearly, wild type obviously becomes this fragment. Even a single point mutant seems to already have an impact, not very much. It's similar to the wild type. Creating N-terminal, so everything is gone. You can see that no tethering happens. So you see the quantitation of it, right? So we know tethering happens and it is binding to a lot of other things. Our question was how? Even now, can it bind to the cardiolipin in vivo? How do you show it? So we did the experiment with TFP VAC31 and NAO staining for cardiolipin. You can see a beautiful colocalization. So cardiolipin and Mycobacterium tuberculosis, VAC31 actually bind in vivo. In vitro they bind, in vivo they bind. What happens when VAC31 is not there? You can very clearly see when VAC31 is depleted or even when it is overexpressed, the beautiful cardiolipin localization that you see, pole, septum, pole. When you overexpress, it's a bit all over. When you deplete, it's an all over. So what is the meaning of that? The concentration of VAC31 matches as far as maintaining the correct placement for the cardiolipin. Now, which is what we don't know. Most probably cardiolipin is what brings VAC31 to membrane. And there it interacts with other things. What is the impact of tethering activity? I don't have tethering activity. There is no rescue inside. So we put an additional copy of it and we show that if without tethering activity, you do not get the rescue, right? So what we showed over a period of time is loss of complementation due to the lack of the membrane tethering activity is basically actually compromises its ability to rescue. Is it because it is whether it is interacting with interacting partners or not? We had so many novel interacting partners. One of the questions of reviewer is, are they really binding or you're getting just like that? You have to validate. So we did it with 4 different proteins. This is not the best gels. We have now very good ones. What we did is we expressed them separately in E. coli and in Stigmatis, put them together and did a His pull down. And after doing a His pull down, we plotted with flag and His. And what we showed is that you can very clearly see that wild type and the N-terminal deletion both are coming down, and interacting partners are binding to both of them. And we did this with 4 interacting partners, and all of them are interacting with the C-terminal region. N-terminal region has nothing to do with protein interaction. So it appears to me the N-terminal regions have a role in tethering. C-terminal regions, which form a dimer, are having a role in interacting with proteins. Based on this, we propose this hypothesis that you have VAC31 is a tether. Scaffold is the earlier word, tether is what we are saying, which hold on to the lipid, both ends, and the proteins are interacting in the middle, forming complexes in the right places, which are necessary for the functionality of the cell. And that is why you take it out, this tether activity goes away. VAC31 is gone, cardiolipins move around a bit, and proteins move around a bit, eventually causing a situation where you end up getting cell death. This is our current hypothesis, and the work is almost in the final stages. It's a dimer and a tetramer. In fact, it's done by others. The good part of VAC31, there is enough literature. And the bad part of it is there is enough literature. So you have to find ways to go around and mix and match. So when you show something interesting, it better be novel. Otherwise, you can't publish easily. But since there is a lot of literature, you can spin stories around existing literature and what you have. So this is where we are. And in addition to this, what I've showed as work, there is a lot of work that is going on in many of the projects in the lab. Again, given my schedule, they see me either in the morning 8:30, or they see me after evening 6 o'clock. But they are working on their own, very dedicated lot. Given that I'm able to still do research in addition to what I do. Thank you very much. Sure. No, not the back. I moved a lot. The PKNG is a kinase. It phosphorylates a protein called GARE, which is a substrate. And the GARE then actually, GARE binds to a bunch of proteins. Phosphorylation is a negative determinant for binding. So if you don't have the phosphorylation, it is binding permanently. You have phosphorylation, the binding comes off. So the TCA cycle becomes more robust. That is PKNG. Yeah. No, it's these are simply peptidoglycan is the ligand. So peptidoglycan synthesis for all of you starts off. You may have heard NAMNAG, N-acetyl acid, N-acetyl glucosamine. So peptidoglycan is basically made in the periplasmic region. Correct? You have NAMNAG connected to a lipid that's called lipid 2. So lipid 2 is completely intracellular and it flips out into the extracellular domain through function of certain proteins. When it flips out, it is sitting in the periplasmic region. What we showed is that this lipid 2 is the ligand for people. So it's not anything to do with the host. A pathogen mechanism to control its own cell division. Okay. And this lipid 2 then has what it does is that it transfers this particular part of the NAMNAG and the peptide, pentapeptide that is associated with this to existing chain, thereby increasing the length of peptidoglycan. So lipid 2 concentration in the cell is very low. It's only around 200 or 300 molecules at any point of time. And it is actually considered, if you can inhibit lipid 2 synthesis at any point of time or inhibit lipid 2 per se, it is considered a fantastic drug target. You inhibit any of these processes, you can see PKNB seems to be controlling the process. You lose PKNB or the control of this process if the lipid 2 binding is abrogated. Once you lose the control of the process also, it is done. So it's a homeostasis issue. It's an issue. What happens is cells want things done when they want it done. And they have many checks and balances, such as, the protein expression, localization, and, what we call as a liquid-liquid phase separation. All these things are mechanisms used by the cell to make sure that they make things when they want to make, not otherwise. And anytime it goes into the host, there is a stress. So when there is stress, for example, latency, people have shown, again, not our work, that PKNB is gone, degraded, not present even at a protein level. Transcription goes on, but PKNB gets degraded. People do not know the mechanism yet. Most probably it gets modified and gets degraded by ClipX kind of pathways, but nobody knows. So what is happening is, in response to what host is putting on the bacteria modulates how these things work. So that's the response, right? Not much, 60%. They both are. In fact, I believe, this is very, they are distinct targets. More than anything else, I believe, I'll tell you the problem with kinase. It's easier said than done, but there are 800 kinases in the host. Okay. The ATP binding loops are very, very, very similar. The N-terminal region of every kinase. The ATP binding domain is very similar across the board. So getting a specificity, because most of the inhibitors that are available in the market, whether it is Gleevec or anything, they are still ATP binding domain inhibitors. Most of them inhibit the ATP binding or ATP interaction with the kinase. Okay, so you have to get an inhibitor which gives you at least 100-fold higher activity for PKNB compared to everything that is present in the eukaryote. Very specific. Now, if you can get something that is specific to both, and there is another barrier that goes through that fantastic thick cell wall. If you can get through this, it's a terrific task. Yeah, it is solved now. The crystal structure is also there. I think there is a lot of people have worked on. No sheets. None. It's like a long thing, and the N-terminal region is different. So what we are thinking is that the N-terminal region binds the hangs. I consider it as like hanging onto the bus, and the middle part is what is essentially providing support to many others. Yeah. Yeah. What I'm calling it as feather, you can call it a length. Yeah. No, I believe what we think. I mean, I have no particular proof. That phosphatase is also localized, right? So kinase activity is kept in check, right? Not low. But what is necessary for phosphorylating many substrates which are also localized there. Many substrates are cell division substrates and cell wall synthesis substrate phosphorylation is also has to happen in the right place at right time for their activities to be controlled. What is phosphorylation? It's a regulatory mechanism. You actually hyperphosphorylate or you hypophosphorylate. In both cases, regulation is lost. So the PKNB and the phosphatase are localized to the pole. They also have the ligand. So the whole thing is controlled. For the moment ligand mechanism. Phosphatase is still localized. Kinase is not anymore getting to phosphate. I don't have a proof. No idea. I mean, but it seems to be doing it both in vitro and in vivo. Okay, but cardiolipin is a major component of mycobacterial cell wall. So maybe it has ultimately confirmation. I realistically don't know why only this and why not, because there are a lot of negative charges in PE. Okay, and in PG is also pretty much part of the membrane, but it seems to be going only for cardiolipin. But it's a confirmation issue.
So these are various constructs where you have shown that they are essential for survival. So if you have tried to immunize this bacteria and look for when they achieve a kind of level threshold level in the mice and then you induce and bacteria dies. So what kind of protective immunity?
I think immunity doesn't come into play in many of this work, other than in case of some secretory proteins. Because here we are talking about the fitness of the bacteria itself, right? The bacteria is not able to manage its own metabolism, whether it is PKNG. PKNG is known to be secreted, and it is also known to have some role in the host. But I believe that major role of both PKNG, this is my belief, okay? PKNA and PKNB is in the pathogen itself. Pathogen needs these proteins, and when you start depleting these proteins, the pathogen's own functionalities are getting compromised. Metabolism, or cell division, cell wall synthesis, these are getting compromised and nothing much to do with the host here. However, if suppose I were to be working on some of the other aspects, for example, in work that I have not presented today, is about showing that Mycobacterium tuberculosis actually secretes around the 300 proteins. And when secretome actually regulates the ATM and AKT kinase in the host. So what they do is secretory proteins go to cause a minimal DNA damage. This minimal DNA damage is sufficient for ATM to stay active. So if when ATM is active, it phosphorylates another host kinase called AKT. AKT is a kinase that is involved in apoptosis, regulating apoptosis. A phosphorylated AKT inhibits apoptosis. Dephosphorylated AKT helps in apoptosis, go towards. So what we showed is that the pathogen actually does enough damage, reasonable damage, but not killing it, sufficient to keep ATM active, sufficient to keep AKT active, so inhibits apoptosis. So you're going towards necrosis, which is good for the pathogen. So what I'm saying is that pathogen has two distinct parts. One is it is controlling the host. Second, it is also trying to control its own metabolism. Like it is inducing cysteine biosynthesis when the stress comes. Why does it need to do it? Because if you have cysteine, more cysteine, you will have more mycophenol, which is equivalent to glutathione. So if you have more mycophenol, oxidative stress coming from pyruvate oxidase or from phenol, nitric oxide synthase, both of these things can be taken care of. You have less cysteine, they will die. It's like it has two different work when it goes inside. One, rewire itself. Second, modify the whole. Both it is and very effectively. The latency is basically a scenario where the oxygen level in and around the bacteria are low. So, and if you are going to eat like this, I take bacteria and I completely take away the oxygen, to die. So latency is a slow adaptation. It's not a one-shot adaptation. When the oxygen levels start going down, pathogen continuously adapts. It does it by decreasing its metabolic speed. So when it is latent, the division is much slow. Okay. So what happens is TCA cycle and central carbon metabolism, even the other ones, lipid biosynthesis, everything slows down. So some of the proteins like CKMB get degraded. So all of a sudden, cell division is not happening. The levels of the lipids go down. Levels of, a peptidal ligand becomes more meshed up. Meshwork goes up. It becomes resistant to things getting in. So you once it is latent, you cannot kill it with rifampicin. You cannot kill it with isoniazid. You need ethambutol, which is actually works during that time. Or pyrazinamide, I think. So pyrazinamide, not ethambutol. Pyrazinamide. You need some of these things which are very, very essential processes that will survive. Everything else slows down to such an extent that even drugs don't work. No, they are not a source of infection, but they're persistence in your body. I mean, they're not going away, right? So when they get an opportunity, they're back up. They're not infecting anybody anymore, but they're there. So, our body, when you are 20, it is not the same as when you are 60, right? You actually end up getting arthritic pain, right? If you have arthritis, you have to go and take the anti-inflammatory medicine, right? So all these things are fantastic for the patient. That is what I was showing. The protein kinase G, for example, seems to be essential for latency establishment. Means if somebody comes to the clinic with a TB, we can give them anyway the anti-TB ATT therapy, but if we can also give a PKNG inhibitor along with that, we should be able to eliminate the latent bacteria. But we have not worked on it further. The result is out there that controls the TCS. They are there in Streptococcus pneumoniae, they are there in some of the other bacteria. They are not very concerned that way. But the way they work in each bacteria is slightly. So for example, Corynebacterium glutamicum also has a PKNG, which works in a very similar way. But Corynebacterium glutamicum has a different kind of a function, a different kind of a system. So what they are used for is slightly different. For example, the AOSR which I was showing, it is there in smegmatic, but it doesn't have the N-terminal region. Seems to be working differently in pathogen versus non-pathogen. So there are very, very minute differences because pathogen grows much slower. It has its own mechanism.
So, coming back to this that one story, you have shown the clusters of 45 So usually, if you look into mammals, there's a lot of tribes, right? Followed by EF1, EF2, you have a clusters like problems. So here, particularly, you are restricting towards cardiolipin.
No, cardiolipin is the binding. We have not looked into the composition of the lipid, but we assume that to be everything. We have looked at the lipidomics we have done. I have not shown the data. It is a combination of multiple things. And the lipid composition doesn't drastically change. The lipid bodies are not made of cardiolipids. They are made of everything.
So then usually when I look into it, liquid phase separation. So here, what do you think the Ragman tethering is required for that?
No, I didn't say anything about lipid phase separation in the context of activity. I was saying that in the context of sigma A. But in the context of VAC31, what we notice is that it has two distinct activities. I mean, activities, I won't call it, two distinct functions. One of them is its tethering, and that is through cardiolipin, and that is through the N-terminal region. Second, it is interacting, and that is through the C-terminal region. So we are essentially assuming that it is kind of holding on to lipids and acting like a bridge for the factory stage. Hypothesis. If I don't have, when we complement it with a VAC31 which does not have the tethering region, okay, it is dying. So it's not cytoskeletal. Cytoskeleton is a different scenario. Here we are talking about direct binding to the lipid. I don't know the answer for that. I mean, whether in other pathogenic bacteria, whether it is asymmetric or not, I'm not looked up. I mean, if I would have known, or I would have to look it up and give you an answer. So it's a way for bacteria to go towards persisters because you have smaller cells which are metabolically at a different level compared to the bigger ones. It is a housekeeping sigma factor. 50% of the genes are regulated by 50%. However, let me tell you, housekeeping sigma factor it may be. There are other sigma factors which are conditionally important. Now, we have to do a chip sequencing to show that sigma A promoter binding changes in stress conditions. We have not done that. I'm talking about stress conditions. Normally, it will bind and control quite a lot of promoters. But how many of them are directly controlled? How many of them will be controlled under stress is a different question, and we have not yet attempted that. We plan. We believe that it is interesting, you know what, I can delete 132 amino, right? It will work. But if I do phosphorylation mutant in the 70th amino acid, that already compromises the function. So B. Gopal in his lab has shown that there are regions in the N-terminal which are required for liquid-liquid phase separation. So if you do not have this, there is no liquid-liquid phase separation. The migration with when we did a GFP labeled recent data, unpublished altogether, is that if we take a GFP tag sigma A, it gives you a dot kind of a localization. And if we take N-terminal deletion, which is tagged with GFP, it is all over. Smooth localization. So liquid-liquid phase separation is gone. The movement N-terminal region is gone. So it seems to be involved in keeping this liquid-liquid phase separation, which essentially means it may be interacting with a unique set of proteins. Don't know. That region is indistinct. Essentially, the liquid lipid accumulation is not specific to VAC31. You can put a cell under stress, you have lipid accumulation. Quite a lot of times in the stationary phase, you see lipid accumulation. And this kind of exactly the same thing as this can be seen in stationary phase. So, you know, in the context of cell, I believe lipid accumulation is due to one, factor is moving around. Second, it is a stress response mechanism where you are storing energy in the form of lipid. It happens even in our body. Wherever lipid accumulation is sometimes a stress response. So, you know, for example, even in granuloma, quite a lot of our cells tend to accumulate lipid. And when they accumulate, they're called, foamy macrophages. Under those circumstances, what NTB does is switches its interest to using foamy macrophages to metabolize from there. So you're cutting off the source of the food then it does. You start using the foamy macrophages as food. I mean, lipids are food. It's a pathogen that is evolved with us. You throw something at it, it finds a way. Yeah. Slow, very slow. Because the speed at which a cell division happens is once in 20 hours for Mycobacterium tuberculosis. The latency is much, much, much lower. Nobody realistically exactly calculated, but it is lower. But it has to divide. The division is not linked with stress. No, age factor is a different one. Age factor means our host immune system becomes weaker. Yeah, it is kind of, see, your body is fitter. Okay, things, I believe it's homeostasis, as I said. Your body is infected, it found a way to contain it. Now, person who's actually having a latent infection is really say diabetic. Scenario changes, metabolism changes, and that will eventually have an impact on the latent, the granulomas also. It's a scenario where host and pathogen have come to some kind of a terms of agreement, right? Anything changes, no, didn't do it. What are you asking? We haven't done it, but we expect it to be. The cell wall composition is much, much more robust in pathogens that are coming from a host compared to the pathogen that is grown in the lab. For example, there are certain lipids which are not seen in the PDMs, they're called. They're not seen in a lab grown bacteria, but you take a clinical strain, it has a memory. So you grow now that in bacteria, you see the lipid composition, it changes. Yes, it changes. That part has been shown, but we have not done. Yeah. See, there is always a path ahead. I can pursue it, but I need a fantastic medicinal chemist, and I need people, industry also, to work with me on this. Otherwise, you've seen the valley of death, you're aware of it. You have a lead, you have this some interesting molecule, it never sees the light of the day. For that to happen, you need a team with a very different set of expertise. What I do is only one end of it. I need at least two other partners who are very motivated and good at what they do to work with me for me to take it further. But even if I don't have a team, if anybody else anywhere wants to take it further, I'd be more than happy to help. Mm. Doable, but we need a fantastic team. I mean, basically, this cannot be done without the industry and medicinal chemistry. True, it was considered that way. It is changing. I mean, I would say again, comes back to the fitness of the person. Everybody is getting exposed, right? Now, who develops a disease depends on the person, right? Now, there is a problem of the rich also, where they have a lot of lifestyle diseases. Obviously, they're not the fittest around, right? Poor have a problem of nutrition, so they're not the fittest around. So earlier it was considered a disease of the poor because it's a nutritional part. People who are not properly, they are very skinny, realistically are not getting nutritious food, obviously are more prone to infection. But on the other hand, if you're having a lifestyle diseases and you are consuming food which is processed, too processed, and you're putting on a lot of getting other diseases like hypertension and diabetes, you're also prone. So it's a simple concept of what is considered rich, what are they doing with that, right? A fit guy, a fit person, fit woman, fit man, they'll be fine. But point is, if you also have a bad lifestyle, doesn't matter what your economic strata is. We are working on it. Pathogen composition from a genomic angle, we are working on, but pathogen composition in the context of lipids, doable, we are not working on it. And pathogen composition in terms of transcriptome, we hope the memory sustains at least 2 rounds of in vitro culture where we can actually make it. So certain aspects we are working on, not solo. It's a huge collaboration called TB Genomics project we have, which involves almost 8 or 9 institutions, because you need to get clinical samples, you need to get repositories, you need to get the sequencing done, and NVMG and CCMG are involved in sequencing components, and then you need to get the analysis done. NIA is involved in analysis, sites are involved in sample collection, ICGB is involved in repository. Doing this kind of work requires multi-institution without worrying about where the publication is coming from and who's going to be the author.
I know. We all do this. That's what I'm finding. No, I'll get it put somewhere.
On behalf of organizing committee, I would like to propose vote of thanks. First of all, I would like to thank, with due respect and gratitude, Dr. Nandpuri for not only spending his valuable time, but also giving a very commendable talk on this very important zoonotic infection. And I would like to thank our director, madam, for guiding us and all her support and encouragement for organizing this event in such a very short time. And I would like to thank all my scientist colleagues and students who have, supported and also enthusiastically participated in this event. Finally, I would like to thank the administration staff or senior manager, and finance and also staff for all the help and support in successfully organizing this event. So thank you, everyone. So we have refreshment and poster session outside. Before that, we can have a group photo.